Hello everyone, I know the video cut off quickly again, so let's keep going. Okay, so we have these parts here that we're going to go over. So we have helicase, which unwinds the DNA. The helicase is this right here in the drawing, the orange, okay? That just is unwinding our DNA, right? Because we know it's in a double helix. The helicase is unwinding that DNA. Then we have single-stranded binding proteins that bind to the unwound DNA already to prevent it from re-annealing itself. So the single-stranded proteins are in blue and they attach to the unwound DNA to stabilize it. Then we have DNA topoisomerase, which alleviates the stress. Like, you know, if you have something that's really wound up and let's say you start unwinding on one end, there's parts of that is still going to have a lot of stress in it from being wound up. So this topoisomerase will alleviate the stress as well. So then we have, so these three things that help with our replication really help to open up our DNA strands so that we are able to get in there to do the replication. So then we have primase, which synthesizes RNA primers. And the RNA primers are used here, and we need those primers to begin the replication process. So once the primers are put in there, then we have, and primase synthesizes these primers, and then we have DNA polymerase 3, which synthesizes a new strand, right? So it's looking at the template strand, the template strand, and is figuring out what nucleotides we need to put here to start replicating this DNA and making a complementary strand. And then we have DNA ligase, which will steal together the Okazaki fragments. So as we mentioned before, in the lagging strand, you'll have fragments because DNA is built from 5' to 3'. So you'll have DNA ligase that will kind of be in between those fragments and connect it together. So those are the main parts of this replication fork right here. What I was going to mention before was about the 5' and 3' end and knowing, why there's a lagging leading strand. I know I just touched on it, but there are so many videos that will go into so much detail about it. So if that's a concept that you don't completely understand, please go watch another video as well. But that's just to, refresh and reintroduce the concept here because we definitely need it for this. Okay, and finally, let me go on this side. Finally, we have just a little comparison between prokaryotic and eukaryotic replication. So in prokaryotes, the origin of replication is only one origin per chromosome, whereas for eukaryotic cells, they have multiple origins per chromosome. For synthesis, we have DNA polymerase in eukaryotes that does the synthesizing, and for prokaryotes, they have a bunch of different ones that contribute to the synthesis of the complementary strand. Different polymerases, that is. Then we have to remove the RNA primers, right? Because the RNA primers can't, stay in there forever. They are just indicating this is where, we can start this replication. So to do that, in prokaryotes, DNA polymerase 1 does this. It's 5' to 3' exonuclease. And in eukaryotes, RNase does this, also 5' to 3' exonuclease. Okay? For removing supercoils, as mentioned, for eukaryotes we have DNA topoisomerase, and prokaryotes use this as well, but they also have DNA gyrase, and that is an important distinction between the two. And finally, telomeres. Telomeres, I know we talked about cancer as well, and when we talked about the cancerous topic, but the telomeres are basically at the end of a DNA strand, and they're there so that you're not actually losing DNA, because when you get to the end of a strand, it's difficult to, you have all of the factors binding here and DNA, the DNA polymerase and all those things at the end of this strand here. You won't be able to get this last part of the DNA and actually replicate it, which means you're going to lose DNA through replication. However, we have telomeres that are placed at the end of a chromosome so that you don't lose any real important DNA, but that you just have extra sequences that you know, we know that the purpose of them is to be cut off. And I do think we talk about this in another chapter. I think I draw it out and things like that, but just in case I didn't, that's what it is. But so for eukaryotes, telomerase is synthesizing those telomeres, whereas that is not applicable for prokaryotes. So I hope you enjoyed our video. I hope you learned something. I hope you understand all of this because it's super important. But we will see you in the next video. If you have any questions, comments, put them below.